So I'm Ashley Brodell, and I'm the Miller Fellow in Behavioral Neuroscience. And see this picture right here? That's actually inside the brain, which is a really awesome thing I got to see every day during work. So the Blumberg lab is a lab at the University of Iowa that looks at the neural development in primarily rat brains, sometimes mice, and they've even actually started to look at humans as well, human infants. And the overall theory is that infants sleep, they move around a lot. That's dreams, right? No, that isn't the theory that they're looking at. It's actually reflective of neurodevelopmental processes. So my project in particular, I looked at the cerebellum, and basically it's a brain region that's important for sensory motor integration and proprioception, which is basically like how you know where your arms are in space. And cerebellar development is especially good to look at in rodents because this region develops after birth. So I looked at ERBB2 knockout mice, which is a gene that's necessary for adequate muscle spindle development, which are located in the muscles, and it's useful for proprioception. And the general hypothesis is that mice with ERBB2 knockout, which means the gene isn't functional, basically, okay, so, sorry. So basically, yeah, they would show less climbing fiber innervation than wild-type mice. And here is a bunch of pictures of all the stuff that I did while I was there. So the first one was polymerase chain reaction to see if the mouse is actually a wild type or a knockout. And then right here, this is a microtome, and I sliced brains using that. And it was kind of frustrating at first because we had to mount the brain slices on the slides, and that was a bit of a challenge to learn, but by the end I was a pro, which was very awesome. And then we did immunohistology, which basically stained the cells with antibodies, which produce images that look like that. And then before those images were produced, Nick took the slides to a confocal microscope. This is right here, and I got to observe how the pictures were taken. And then afterwards, I used image J and outlined all the cells with that, so then we could analyze the data. So in short, the results show that proprioceptive feedback from muscle spindles is important for cerebellar development. So this is age 15 days after birth knockout mouse, and then this is a wild type. And as you can see, a noticeable difference in size. So basically, I learned a lot about a brain region that's not as often talked about as much in psychology classes. Is underemphasized, I think. And I also learned a lot about different research methods in the field that I'm very interested in going into. And then I also was lucky enough to be able to sit in on lab meetings, which was a good way to see how science works and how it's all put together. And I also learned how to present my ideas confidently and to not second guess myself as much. So the current impact is that I learned about the inner workings of brain research, because it's so easy to say, hey, I want to do research, but then when you actually do it, it's much different. And it's just like the workings of the project. And I also learned that I'm very interested in researching sleep farther. And in my last class, I even expanded upon that and did a presentation, which was really cool to learn more about. And I can definitely reflect on this experience in graduate school applications. And I also was able to network with research professionals and learn about what they do and how they do it. So basically, my suspicion has been confirmed. Research is where I want to be in the future, so that was a really awesome thing to learn. And I have a better idea as to what type of labs to look into when I'm looking for different graduate schools to apply to next year. And I was also challenged both personally and professionally, and it really helped to helped my growth as both a student and a professional, just in any way, even personally. So I would like to thank my site mentor, Greta Sokoloff, and Nick Sadler, who helped train me with everything. They both went up and beyond my expectations to help integrate me in the lab, and they found a really awesome project for me to work on in which I got results. And I would also like to thank my faculty sponsor, Carolyn. She was always there whenever I needed to talk to her, but unfortunately she couldn't come because she's in Europe right now, which is cool. And then I would also like to thank everyone else who has helped me, such as Sue Astley, Jason Napoli, Rebecca Sullins. They've helped me out a lot, both before and after my fellowship. So, yeah, thank you guys for coming.